All right, this discussion on chapter 15, biosynthesis, is going to be quite truncated. We're going to focus on just a couple main points. Everything else will be important if you want to go into a course like microbial phys or microbial ecology and potentially diversity of prokaryotes as well. But we're just going to hit a couple high notes here. So here's the learning objectives and key terms all in one. And we're going to start off just by reminding you of these terms. So we are talking about now anabolism, the building side of metabolism. Remember that catabolism breaks down molecules, usually to extract energy from them. Anabolism takes molecules, uses energy, and brings them together to build a lot of our polymers, like fatty acids, like DNA, like our proteins, things like that. So, another word for this is biosynthesis. We're synthesizing or making the molecules of life. And overall, biosynthesis requires three main things. First is the essential elements. All organic compounds are made of carbon, hydrogen, and oxygen, and then we add on things like nitrogen, sulfur, phosphorus, and whatever else we need. But those are the essential ones. And then we need a reducing agent, something like NADPH. So we're going to be building up molecules by oxidizing them, and to oxidize something, you need a reducing agent. And where do we get NADPH and the energy from ATP? Where do we get this from? We get it from catabolism. So we break down molecules to get energy and then use that energy to do this biosynthesis. So energy is the last requirement of this. Um, if we take a look at this image here, you are not going to be responsible for knowing all of this, but we want to get an idea of what we're looking at as a whole here. So one of the key intermediates of both metabolism, or of both, excuse me, of both anabolism and catabolism is acetyl-CoA. So acetyl-CoA can go into the TCA cycle, or it can be used for gluconeogenesis, which is creating glucose. It can be used for the synthesis of fatty acids and lots of other stuff. And so this whole pathway, if we notice too, it's also including things like nitrogen fixation and nitrogen assimilation, because we have to take that um, super stable form of nitrogen gas in the atmosphere, and break its high energy three carbon, or yeah, the triple bond, excuse me, the triple bond between the two nitrogens. We have to break that down, and it requires a lot of energy. But once we have the organisms that can do that, we then take that nitrogen and assimilate it into things like our amino acids and our proteins, which require a significant amount of nitrogen. They need those amino groups to make their amino acids. If we are going to be building up all of these molecules, we require many different substrates, a lot of building material effectively. And where do we get a lot of these from? We get them from glycolysis in the TCA cycle. So we take a bunch of the intermediates from those steps and funnel them into these biosynthetic pathways. So an example here, if we're looking at the TCA cycle, one of our intermediates is succinyl-CoA, and this molecule can be funneled away from the TCA cycle and be used to create many amino acids. It can also be used to make vitamin B12. Humans don't do this, but bacteria do this. And then we have glycerol 3 phosphate. That's going to give us the backbone for a lot of our lipids, that glyceride backbone. Then we have pyruvate. This is going to be the backbone for many different amino acids. So glucose catabolism and the TCA cycle are both reversible, and they create a lot of intermediates that are used in both metabolism, or keep doing that are used in both catabolism and anabolism. Now, one particular reaction that we're going to just mention We're not going to get too deep into this. That's more a microbial phys class topic. But if we want to take substrates and actually make glucose from it, we call that gluconeogenesis, and it's basically reversing the process of glycolysis. Then all of this requires significant energy expenditure. So a lot of times these enzymes will do those coupled reactions. So they will couple a synthesizing reaction, one that's doing anabolism, with one that's catabolic, with one that's breaking down molecules to release energy. And overall, the cost of this is incredibly high, such that we're even starting to talk about a genomic cost on the microbe. So if we need to build absolutely everything in our cell, we have to have the genes to make the enzymes to do that. Think about the trip operon that we learned about. This had 5 genes in just this one operon, and the only job of those genes was to make one amino acid, tryptophan. And remember that the larger the genome, the higher cost it's going to take to maintain that genome. We're going to have enzymes that have to regulate, or proteins that have to regulate, enzymes that have to copy and transcribe, and all of those just add to the energetic cost of being alive. So one way that cells kind of balance this is by figuring out genome loss and cooperation. So they don't necessarily have to make everything on their own. Humans don't do this. We don't make a lot of amino acids on our own. We have bacteria that live in our gut that help us with that. So a lot of times organisms will cooperate with other types of cells and form endosymbiotic relationships, or with other types of organisms that live nearby. And they can say, well, you're going to make this for me, and I'm going to use it so I can delete those genes from my genome. I can lose them and reduce the size of my genome. So one of these anabolic reactions is the Calvin cycle. And I know you have heard this many, many times. This was part of photosynthesis. It's actually the light independent reaction part of photosynthesis. But remember, this evolved first in bacteria because bacteria were around before there were any plants. So this is done by not just bacteria, or not just plants, but bacteria and algae and all these other organisms, including many microscopic eukaryotes as well. Now, these are the light independent reactions, meaning light does not have to be involved. So a lot of organisms that do this do not live anywhere near light that can be used for this. Light is fantastic because it's energetically costly and we can use light to power and provide that energy, but not all organisms that do the Calvin cycle use light to power it. Just keep that in mind. And another term for the Calvin cycle is carbon fixation or carbon sequestration. This is taking carbon dioxide from the atmosphere and removing it and putting it into the form of biomass, either the structure of the plant or the structure of the organism that eats the plant, or it goes into the soil where we have a big carbon sink. Now, this is also known as the reductive pentose phosphate cycle, but very few people actually use that term, but it is a little bit more descriptive about what's going to be happening. But the Calvin cycle is a perfectly fine name to know this by. And like I said, it's responsible for nearly all of Earth's biomass. So all the mass that we see on the planet that is organic came from an organism that can do carbon fixation or the Calvin cycle. Now, not every single bit of biomass, however. We're not going to get into those extra things. This is done by many different types of organisms, so oxygenic phototrophic bacteria, algae and plants, those purple bacteria, and some lithoautotrophic bacteria as well. But you do not need to know all of those. So let's look at a quick overview of the Calvin cycle. It's going to take CO2 and water and condense it with a ribulose 1,5-bisphosphate. This is a 5-carbon molecule, and it's going to create effectively glyceraldehyde 3-phosphate, G3P. So all of this is catalyzed by the enzyme Rubisco. So if we look at really what's happening here, I'm going to go to the next slide. We're starting with a 5-carbon molecule, and we're taking CO2 and we're adding 1 carbon to it. This creates a 6-carbon molecule, like something like glucose. And then we're going to take that 6-carbon molecule and break it in half. So now we have 2 3-carbon molecules. These 2 3-carbon molecules go through a series of redox reductions. And remember, when we reduce something, we are adding energy to it in the form of electrons. So we're adding energy to these 3-carbon molecules, and we get to a final product here of G3P. Now, one of these can be funneled out and can be used to make glucose in gluconeogenesis, or used to make amino acids or whatever else we might need it for. It can also keep going and now enter into the TCA cycle as if it combines with acetyl-CoA. Now, the rest of it has to be funneled back into the cycle, because this is a cycle, so it has to regenerate the starting molecule for it to keep going. However, this is a 3-carbon molecule, and ribulose 1,5-bisphosphate is a 5-carbon molecule. So we are down 2 carbons. We need 2 more carbons to be able to regenerate and keep redoing this whole cycle. So every single time we do this, we have to run it 3 times. You see here, there's actually 3 carbon dioxide molecules entering. When we do that, when we run the whole Calvin cycle 3 times, then we are able to regenerate the 5-carbon molecule that we need, the ribulose 1,5-bisphosphate, to keep the whole process going. Moving away from the Calvin cycle now, we're going to just very briefly talk about a couple other anabolic processes. So we can make our own fatty acids, and cells can actually make their own antibiotics. The very first antibiotics that were discovered were created by other microbes, and then we just learned how to take those base antibiotics and modify them a little bit into newer antibiotics. But many of these components are made from repeating units. We've been kind of hitting this a lot with DNA and amino acids, how they are the repeated units of nucleic acids and proteins, respectively. Now, the reason why the cell does this, why it makes a bunch of repeated units and then sticks them all together, is because this produces a lower energy cost. We're going to be able to use the same machinery to pump out the same product over and over and over again, like a factory line, and then just kind of stick them all together to make the larger molecules that we need. And then any modifications can be done by individual enzymes later on. And those are often going to be able to do the same modifications on many different types of molecules as well. So we call this modular synthesis, where we take repeated units and use them to build more complex products that will often have various R groups. So this is like fatty acids and antibiotics. Then there is nitrogen fixation. So this is required to make amino acids. It's also required for the cell wall, other cofactors, but primarily we discuss this in terms of needing to make our amino acids, which build up our proteins. So since this is one of the most stable molecules in all of nature, it requires a huge amount of energy to break that, and we can thank bacteria for doing that for us. So once we have broken down that nitrogen into a form like ammonia, it can then be taken up or assimilated by organisms and then used to create other molecules. So this is just kind of an image of this happening here and the different forms that nitrogen can cycle through. We learned this a little bit in the lab as well, where we looked at the denitrifying bacteria and how the first step is to take nitrite and turn it into, or nitrate, turn it into nitrite. Then that nitrite can be further reduced down to nitrogen or the nitrogen gas that we saw, or it can be taken down to ammonia. And that ammonia is the one that will be taken up and assimilated by cells for biosynthesis. Now, when possible, organisms like to get their amino acids from the environment. They'll use membrane-embedded transporters and bring them in. However, this is highly competitive. Everything needs amino acids to survive. They're energetically costly to make. So if you can make them, it's good to be able to do that rather than being completely reliant on getting them from an outside source. And so most free-living microbes and plants can make all the standard amino acids that they require. So with this, a lot of the skeletons of these amino acids are coming from intermediates of metabolism, coming from intermediates of glycolysis or the TCA cycle. And you can kind of just see that here where we can shuttle specific intermediates away and make the amino acid from them. So pentose phosphate right at the beginning of glycolysis can create histidine. Pyruvate can be shunted off to make valine. Acetyl-CoA can make leucine. And then we have alpha-ketoglutarate or 2-oxoglutarate. This one can create glutamate, which can become proline, arginine, and glutamine. So lots of different amino acids can be created from this one intermediate. Other amino acids have to be made from preformed amino acids, like we can see here. We have to make glutamate first, then from it we can make the arginine, the glutamine, or proline. So that is just kind of a very, very brief overview of amino acid biosynthesis. Read the book if you're curious about more, but we are not going to have time to cover more than that. So I hope this helped. I hope it gives you some peace as you're going into your exam, so you can spend some time studying this as well and not feel like you have to wait till Monday to get the information. Please contact me if you have any questions, and I hope you all have a fantastic weekend.